All right, I'm seeing 10 o'clock on my computer screen here, so we'll get started. Looks like we have a big crowd today. Thanks for joining us, everyone. My name's Noah Newman. I'm an editor with No Till Farmer. And today's webinar with Holganix. Our presenter is Holganix President of Agriculture, Dave Stark. You see him right there on your screen. He'll be digging into the role soil microbes play in the breakdown of crop residue and building soil health. And here's a fun fact about Dave. According to my stats, he has 26 peer-reviewed scientific articles and holds 9 patents. So a very impressive resume. And before we get started, I want to remind you, we're going to have a brief Q&A session when Dave gets done with his presentation. So if you want to type your questions in that Q&A box at the bottom of your screen, it'll pop up on my screen, and I'll relay those to Dave towards the end of the presentation. And we're also recording everything, so if you miss anything, you have to get up and grab a cup of coffee or iced tea and come back. You can go back and watch what you missed. We're going to send the recording out within 24 hours of this presentation. All right, let's get started. Dave Stark, take it away.
Okay, hey, thanks a lot. Thanks everyone for taking some time to learn about residue and microbes. So we'll just kind of get started here. So I just want to start with a different thought, and this is something. So if I think back, like I said, Noah earlier, I've been doing agriculture technology for 40 years, which is kind of stunning. It's a long time. We thought, so when I was at Monsanto, I was in the lab that actually developed the Roundup Ready technology. We invented the gene, or we found the gene, we discovered it. And at the time we thought Roundup Ready might help make no-till easier. And I think it did to a lot of extent. But what I've learned now, really working with soil biology and soil health, is a different way of thinking. I'm seeing that when we improve our soil, we actually do a lot to make no-till also more practical. So, just a thought to plant that we'll see throughout the presentation, that soil health actually plays a pretty big role. Part of it, we'll talk about residue management, and we'll talk about the role of different microbes, about different kinds of products to control residue, what to expect when you use them, and hopefully you learn something, because that's my goal. So I think all of you know that your crop residue has a lot of value. Clearly, you can remove some of it or all of it as animal feed, and that's really worth a lot of money to you. But there's also value leaving some or all of it out on the field. Keeping the soil covered and minimizing erosion is probably one of the top things. But also, all that carbon is helping build the soil, because that's food for biology. So there's a trade-off that I know I talk to a lot of guys about. If we leave too little residue on the field and we have bare ground, again, we promote erosion. Erosion is your number one source of nutrient loss. Of course, you lose valuable topsoil as well. And you all know that you've got soil prone to surface crusting. So you plant a crop. If you get a rain, the crusting keeps it from coming through. So you may have replants and such. But also, the lack of putting carbon back in the soil, we eventually degrade soil and we lose organic matter, we lose moisture. But on the flip side, if we have too much residue in the field and it's still there in the spring, now there's good things. The residue helps keep the soil cool and helps retain moisture. But in the spring, and there's some good data out there from companies like Precision Planting that show too much residue can lead to bad seed-soil contact. The soil warms unevenly, so we get uneven emergence. And their data is pretty compelling, that even a few days and differences in emergence costs pretty significant at the end of the year in yield. And of course, just physically handling all of that residue. But if we go now and look at, you're going to leave the residue on the field because there's a lot of nutrients. And it's true. These are nutrients you've already spent money on in your fertilizer. So if we look at corn, soybean, and wheat, per ton of residue, and let's just look at corn. For every bushels of corn, you produce about a ton of residue. So your 200 bushel an acre corn crop, you've got 5 tons of residue out there in the field. Each ton has 17 pounds of nitrogen, about 4 pounds of phosphorus, 34 pounds of potassium. And you'll see that potassium is really a big part of stalk and stem strength, and some sulfur. So again, there's a lot of money out there sitting in your residue. How are you going to get it released to the field so it feeds next year's crop? Well, it has to be degraded. So what are some of the factors that influence decomposition rates? Well, here's the truth, the bottom line. Without a microbe breaking that residue down, it's called mineralization, into its basic chemical forms, not a single ounce of any of those nutrients will ever become available. So the right question is, what are the factors that influence microbial activity? Because they are totally responsible for decomposition. Well, there's a lot, and I'm not going to go through all of them. We're going to keep it real simple. Temperature and moisture. Just like your crop. If you know it's growing, if you've got in the right temperature zone and you've got some moisture in the soil, same with the microbes. Temperature and moisture. Now, there are other things that matter. So clearly, the more soil contact, the more we can chop or crush residue, the better. Look, it's just like your skin. You tend to not get an infection unless you have a cut, because that's an entry point for a microbe. The same is true with the residue. The more entry points we can make, the more residue degradation that we can get. So I keep talking about microbes, and we're going to talk more about it, but I like to go over what the microbes are. And I do this so that you can look at different products, because I know there are a ton of things being thrown at you in terms of microbials and biologicals, on and on. And so you can understand what each type does so that you have the right expectation of what to expect. Because there's a lot of good products out there. There are some that overpromise and undeliver. They don't have the biology to deliver on what they say they can do. So I like talking briefly about this. So these are the different types of microbes we find in soil, from the smallest, the most abundant, the fastest to divide bacteria and actinomycetes. In a teaspoon of soil, there can be over 1 billion individuals, thousands and thousands of species, frankly, most of which we haven't even identified yet. Then we get to the larger, slower to grow fungi, algae, protozoa, and nematodes. Yeah, nematodes. Nematodes, most of them are actually good for your soil. Everything here is important for the soil health. And I tell you, this is the way God designed soil to work. And I say that if you've heard me talk before, I'm very deliberate. 40 years ago, as a Ph.D. molecular biologist, I was an expert, and I suppose still am, in evolutionary theory and all that. And I would have told you, isn't it amazing what millions and millions of years of evolution have done to build all this? I can tell you today, following the data, this is not because I came to faith later in life. I always had Jesus as my Lord and Savior. So that has nothing to do with that. It's following the data. The data supports that these are all designed. And mathematicians, I found, have been laughing at us molecular biology evolutionists for decades, because what we propose is, their term is absurdly impossible. So when I say God's design, I mean quite deliberately this. And we're going to show you this. We'll do an example in nitrogen. So here's a picture in the top. These are bacteria and actinomycetes. These are representative pictures. The top is bacteria. That's a close-up of actinomycetes. They're really not that much bigger than bacteria. They come in different shapes and sizes. Bacteria are important in soil, just like all the other microbial types. They're good degraders. However, they're better degraders at things like manure and things that have nitrogen. They do not break down crop residue very well. They don't have the right enzymes. They're not as efficient. But there's a lot of things bacteria do. Because they are the smallest, the fastest to grow, they get the first bite of every nutrient in your soil, whether that's fertilizer, whether that's a degrading corn stalk, whatever it is. They are generally starved. And when food is there, they grab it. That's not a bad thing because they help minimize loss of nutrients. They immobilize them in the soil. Now, when they eat, they tend to release their carbon as CO2, so they don't add a lot of carbon to soil. They absolutely dominate tilled ground. And I'm going to do more, in my webinars in the future where I talk about soil structure and how all this happens. I'm not going to divert and get into that now, but they dominate tilled ground. But here's a concept that I want us to think about, because it's going to be a theme throughout my talk. It's carbon to nitrogen ratio. For every nitrogen atom in a bacteria, there's about 5 carbons. As a biologist, I can tell you these are nitrogen hogs. They have an incredibly high nitrogen requirement. Okay? And by the way, most microbial products that people are selling out there contain just bacteria, and usually just a handful of species. And the reason is, frankly, as a manufacturer, they're easy to grow, they're cheap to grow, they're easy to stabilize. So they work well for manufacturing and distribution. They don't always reflect what soil needs because most ground already is bacteria dominant. Now, fungi, the next class, these are the best degraders of crop residue. And we're going to go into some detail with pictures why that is. So fungi aren't those individual little cells that are microscopic. They form these long hyphae, which form mycelium. You see all that white feathery material? Those are the degraders of organic residue. They eat bacteria. They're really good at unlocking nutrients. They're carbon rich, so they actually add carbon to the soil. When we till, you can imagine how we break up all that white hyphae. So fungi live longer than bacteria, but they need a more stable environment. So when we till, we disrupt the fungi and we explode the bacteria. We knock everything out of whack. Now let's go back to our carbon to nitrogen ratio. For every nitrogen, 15 carbons. So 3 times less nitrogen than bacteria. And by the way, only a few microbial products contain fungi. How you can tell? If it's a powder, it's probably just bacterial spores. It could be some fungal spores, but the shelf life isn't that long. If it's a liquid in a jug, it's tightly sealed, you put it room temperature on a shelf, it's probably just bacteria. It's because fungi and the other microbes that you want in soil all need oxygen. So if they're alive in that jug, they're growing and consuming the oxygen, they're not alive anymore. So the last ones I'll talk about are the protists and nematodes. So protozoa, amoeba, they're larger than the bacteria. You don't hear much about them, but they're really important because they're predators. They eat everything. They eat each other, organic matter. A single protozoa or amoeba can eat 10,000 bacteria a day. And nematodes, again, are predators. That's actually a picture of a nematode eating another nematode. That's a soybean cyst nematode that's being consumed by a beneficial one. So these guys all need oxygen to live. And remember, I said bacteria are most plentiful. They lock nutrients in your soil and they tie them up. These guys eating 10,000 bacteria a day, their carbon to nitrogen ratio is 1 to 30, 6 times lower than the food they're eating. So what's going on? It's like they're eating a steak meal every meal every day. And so, not to be indelicate, but you think of urea, that name comes from the waste that we get rid of, the liquid waste in our bodies. It's no accident that urea and urine sound the same. That's us, who also have a 1 to 30 carbon to nitrogen ratio, getting rid of nitrogen that's toxic. That's what the protozoa are doing, and the amoeba and the nematodes. And what do? They eat bacteria, they spit out ammonia and nitrate. What form do plants take up the best of nitrogen? Ammonia and nitrate. Like I said, this is an intricate, elegantly complex cycle. that all works by design. So now that we know carbon to nitrogen ratios, and we know fungi actually secrete the residues that are required to break up residue, what am I talking about when I say crop residue? There's three main biologically made polymers. And I know this is a busy slide. I love it as a scientist, but believe me, there's no quiz afterward, and I'm not getting into the gross detail, but it's here to make a couple points. Cellulose, lignin, and hemicellulose. This is what predominantly makes up most crop residues by dry weight. So if we look at cellulose, and by the way, scientists are lazy. If you look closely, see the H, that's hydrogen. All those O's are oxygen. The C is carbon. But all those lines that just come together, that's us being lazy. Those are connecting carbons, and there's usually a hydrogen off there too. We get lazy, we don't write them. So the first thing you're going to see, cellulose, those are sugars. That's just a long chain of sugar, a very long chain, straight chain of sugar. When you have straight chains, you can pack them real tight together. If you look at that microfibril picture, cellulose, I equate to like the iron rebar in concrete. So think of it, that's the rebar. What's the concrete? Well, hemicellulose is kind of a glue, but it's lignin. Lignin is extremely strong. So when you've got the lignin concrete, which is really hard to degrade, surrounding the cellulose rebar, that's why you can get a 100-foot tall tree standing up to a 90-mile-an-hour crosswind and it doesn't fall over. It's extremely strong, extremely hard to degrade. Now, we talked about carbon to nitrogen ratios. N is nitrogen. Do you see any N's? There aren't any. This is carbs. So our crop residue is a carb-heavy diet. So what does that mean? So if we look at different products to break down crop residue, or if you have a field like most, which are very, very dominant in bacteria with very few fungi, what can happen? So we're asking bacteria that have 1 nitrogen for 5 carbons to eat corn stover that has 1 nitrogen for 55 to 60 carbons, or wheat straw, which is 1 to 80. How do they do it? There's no nitrogen. So they're either not going to grow or they're going to steal it from your ground. So, you've probably all seen yellow corn in the spring, and it can be cases where you fertilized, you do a soil test, there's nitrogen there. It just happens to be tied up inside of bacteria. So bacteria alone not only don't have the right enzymes to break these things down, there's not enough nitrogen. So we really need the other microbial types to keep cycling the nitrogen to make this more efficient. So what are the different kinds of products that are out there to help you break down residue? Well, I already talked about crushing and cutting, and there's different equipment that does that. Again, the more you can break that residue up, the more entry points for microbes, the better chance it's going to degrade. There are some products out there that are just sugars. So it's just food, a quick burst of food for microbes. That assumes the right biology is there. The problem is we know that most ground is pretty bacteria dominant. There are products that are just microbes, and we'll talk about those next. And there's also a lot of recommendations that you might hear, just put some nitrogen down on the residue in the fall. And that makes sense because the residue is low in nitrogen, and even the fungi need some nitrogen so they can grow and do their job. But I saw a study that was multi-year out of Iowa State University, and they said there's actually no benefit in applying fall nitrogen for the purpose of controlling crop residue. And I understand, to me it's a little counterintuitive, but it's a pretty good study, and Iowa State's a pretty good program. So I looked at, a number of different products out there where I could find labels that are being sold to control crop residue. So our product is Bio 800 plus breakdown. So it's something that's fungal heavy, and they're fungi that are able to break down lignin and cellulose. They can get into that rebar and concrete and actually degrade it. But then I looked at these other products. Again, I'm not going into any company's products by name. I'm not going to do that. That's not who I am. So I just list them as A to G. And all of these are only bacteria, except product B. Now, product B could be interesting. The only thing is I can't find a list of what the fungi are. So if they actually have the fungi that form the hyphae, like the penicilliums and such, that could be a good product. So it's something I just would wish I had more information on. So again, another busy slide, but I got a point here. So look at the picture in the bottom right corner. And I know you all see this. I see it all the time. You go in a field that had corn on it last year, some cases more than a year ago. And what do you see? You still see the stalks. But if you look at the stalk, they're usually hollow, because what's happening, and I don't know if you can see my cursor, see that circle, the cross section of the stalk there in the upper left-hand corner? All that white is called the pith. Those dots are vascular bundles, and that dark circle is the rind. It's usually the rind what we see in the field. So what is this? So the pith, which is clearly the largest in volume, it's the lowest in mass. So it's only 11 to 12% by weight of the stalk. It's not strong. It's not dense. It has a decent amount of water in it, so it degrades the fastest. The vascular bundles in the rind, though, are much more dense. So the rind has 3 times the density of the pith. It's very heavy in lignin. That's why, again, we've got that lignin that's surrounding the cellulose rebar that's so long to digest. That's why those corn stalks are sticking around, and that's 75% of the mass. So we don't want to see just the pith. We want to see the rind start to go. So this is a picture, and I'm not going to say who, but this is from the website of a company selling a product to degrade crop residue. This is their picture. It's bacteria only. And look, what we're getting is the pith, which is easy to digest. That's what's gone. That's what I would expect. But look in the middle. You see the vascular bundles. That's the lignin and cellulose. And the rind is got a long way to go before it's degrading. It's the wrong biology. So here is an example when you've got the right biology, a lot of different fungi. So this is 10 days after application. You can see that the stalk is easy to break in half. You can see actually fungi, all that white fuzzy stuff in the right picture, in the right-hand side, growing throughout the residue. That's what we want to see. So again, our product, and use products like this that have fungi, we've got all the biology. So we say 800 plus, over 800 species. We have bacteria because they're important to the whole nutrient cycling. They keep things in the field. We have over 200 species of fungi, though, because that's what the degraders are, and a lot of protozoa and even beneficial nematodes to keep cycling that nitrogen out. Because even fungi need a little nitrogen, we put 2% nitrogen in our product, just enough. It's not going to help next year's crop, but it's food for the fungi, plus some molasses and other micronutrients. So here's a picture. This is this last fall in Iowa. Now, both the check field and the treated fields were all vertically tilled. So that helps chop up the residue. And I want to tell you, and this is, I got to stress, remember I said microbial activity depends on temperature and moisture. This was ideal. So this picture may not be what you always experience with a product like mine, because again, ideal conditions. So I want to stress that so no one is misled. But they had temperature and moisture, and what the grower said is, wow, now I can no-till. Because now in the spring, I'm not going to need to do my chisel plow. I'm going to sell it because I don't have all that residue that I can't plant through. So again, soil health helping to enable us to no-till. This is Southwest Minnesota. This was actually done in the summer. So this is now following a crop, and you can see He's breaking apart the stalks. You can see the fungi growing in there. Something totally different you may never think about is athletic turf. And Noah, you and I were talking about baseball. Same thing. This is a golf course. This is a PGA course in Las Vegas. And he's talking about not just the roots, but the fact that you don't have thatch. And thatch is the grass equivalent of crop residue, where it can really block oxygen flow and really inhibit the growth of the root zone underneath. And so you've got to eat it up. That's what microbes do. They're eating the thatch, so you don't have to ever worry about it. Another benefit in using a product to control residue, if you can get it down in the fall and you do winter wheat or a cover crop, and I do this in other webinars that I've done also with no-till, like I did January at the annual meeting in St. Louis. Microbes help with germination and establishment, the same ones that are going to help with degradation of the crop residue. So here we see, the crop coming up in the fall better using the breakdown product. Sometimes, you're just in a situation, and we're going to talk about this in a minute in terms of when not to use this product. So we've got some guys that just can't in the fall for different reasons. So this is an example. You see that corn kernel in early spring. So he still wanted to control residue, but he planted his corn in 42-degree soil. And look, it's germinating, which is crazy because, 42-degree soil, nothing is very active, but microbes are slowly active. So again, biology is what makes the soil work. So again, you want to really stress that soil health and the soil biology. And this is an example. So in some parts of the country, this is down in the Boot Heel of Missouri, and you go down into Tennessee and Arkansas, you double crop. You follow your winter wheat with beans. So I've got guys there telling me, that's great, but my wheat straw is still there when I'm trying to harvest my beans, and it's a mess. So I want to break down that wheat straw. And wheat straw, remember, is a 1:80 carbon to nitrogen ratio. It's one of the hardest things to break down. This is, I like this picture because I'm always learning. And I tell people, and I will tell you, here in a minute, if you use any microbial product to control crop residue, you're going to apply it foliar. You're going to use as much total liquid carrier as you can stand. 15 gallons an acre is a typical recommendation. We want it wet into the residue mat. Well, this is an example where the field was too wet to get a sprayer out there. So they flew it on by airplane using only 5 gallons an acre. And I thought, that's not enough. You see any microbial product, if it's on the surface of the ground or on the surface of the residue that's exposed to sunlight, UV light is fantastic at killing biology, microbiology. So you want to protect it from the UV light. Well, here I was wrong, because look, he planted his beans 10 days later, and look, there's enough added biology. We got the beans out of the ground faster, and the residue is breaking down. So again, there was enough moisture in the field. I got to sometimes listen to myself. Temperature and moisture, there was enough there that he didn't have to use the same amount of volume, and he still saw a benefit in his second crop. So again, these are fields. I think this is again in Iowa, and I'm going to talk more about this in different webinars and talks I'll give down the road. Look at the soil texture where you've got good biology, and look at the residue control versus where you don't. And just kind of a teaser, I hear so many times about how they don't see clods in the field anymore, how the soil is so easy to work. Even their soil probes go right in where they use biology, and they don't where they don't. And what's happening, not to again get off base, but if you want to build soil structure, and since you're probably a no-till farmer because you're on this webinar, and so you're conscious about your soil, the way we break up compaction, the way we break up the clods and we build texture is a combination of fungi, roots, and their exudates. They produce a glue that takes the little soil particles, the microaggregates, and make the macroaggregates. And then the fungal hyphae wrap them all together to give them stability. So again, the biology with plants is what's building our soil texture and structure. We're decreasing bulk density, better water infiltration, better oxygen penetration. So again, we take soil health as a way to help enable us to do less tilling. So my recommendations, it's all about the biology, and it's fungi, number one, because fungi have the right tools to break down that cellulose and lignin. Remember temperature and moisture. So if you're in a place where you're harvesting after Thanksgiving and it's winter, or winter is days away, don't spend your money. Unless you're far enough south that you get enough sunshine and enough warming in that mat, because it doesn't matter. When it's cold, the microbes go dormant. When it's warm, they get going again. So, I lived 36 years in St. Louis. Most winters, we had enough sunny, nice days with moisture. I probably would consider using a product like this after Thanksgiving. Here in Fort Wayne, Indiana, I would not, because winter comes and it stays. It's too cold. Also, if you're in a place where it's bone dry and you're not even getting enough morning dew to wet that residue mat, you might want to wait till spring or when you turn your irrigation back on. Because again, if there's not moisture and there's not temperature, the microbes aren't going to do anything. They're just going to sit there. So I'd rather you wait till early spring if you have a residue problem. Again, put it on foliar, use it over the top, use enough water, get it into the mat, unless it's really, really wet and you want to fly it on, I guess. Again, nitrogen alone, not my recommendation from Iowa State. That's generally not giving enough results by itself to control residue. You really need biology. The more you can crush or chop the residue, the more microbial entry points. And again, soil contact is important because you've got microbes in the soil. But again, we really don't want you to till. Tilling disrupts the biology we want, the fungi. So a vertical till seems to be okay because you're really not turning the soil over. But when we break open those microaggregates, we disrupt the fungi. We're really going to set everything back in the soil. So, I threw a lot at you, and I threw a lot fast. I would much rather answer your questions. So I'm open to take them.
All
And
right.
by the way, that's my email address. If you'd rather email me questions, please feel free. Happy to respond.
dstark@holganix.com. There it is.
Yes, sir.
Yeah, Dave, we've had a lot of questions pour in throughout the presentation. And again, if you want to ask questions now, type them in the Q&A box, bottom of your screen. They'll pop up on my screen, and I'll try to get to as many of them as we can here. The first one that came in, let me scroll up here. Will microbes survive for long if just sprayed onto surface residue, or do you need to work in digestion products to get them to work?
No, so again, as long as they're not, applied with a really, really low rate, so they're literally on the surface where the sunlight can get at them. And again, remember, they move with rain. So if you've got a rain event, that's going to wash them into the soil and into the residue. When it gets dry, they tend to go dormant. Okay, well, since I'm going to go there, I'll just tell you. They produce biofilms, which is kind of a carbohydrate, watery thing that is between soil particles. So they'll find these biofilms and they'll stay there. So they're actually fairly resilient. You don't need, if you've got fungi, anything else to break down that residue because they produce the right enzymes. And I'm not talking about unicellular fungi, which are yeast. That is technically a fungus, but they don't produce the enzymes. And a lot of products you may have seen earlier in my list have yeast. That's not bad. That's just not the right kind of fungus. You need the kind that produce those long hyphae. That's why I like showing the black and the white, stringy stuff throughout the residue. That's doing the job. So if you've got that biology, that's all you need to use.
Does vertical tillage damage or disrupt the fungi?
Not as much because we're not turning the soil over. So I'm not recommending vertical tillage. I think it really depends upon your operation. Um, there's a quote I saw from Ohio State, and there's some people there that really have some great information on tilling, and I really like it. And they said, in the editor's opinion, the only time you should till is when you're building a highway. Now that's a little extreme because clearly, if you've got really bad nutrient stratification or you've got some serious compaction, roots and biology will fix that through time. But there may be times when you just got to till. But that should be the exception, not the rule.
Next question is, how should we check performance in the field?
So again, remember temperature and moisture. So I've got a lot of people that have used a product like ours, and if just driving by the field, sometimes you go, wow, I can see the difference. Other times you got to walk out there and actually pick up a corn stalk. If you can crush it in your hand, that now you know, because remember, they're mostly eating from the inside out. Because they're protected from sunlight, there's more moisture inside. The outside of a stalk or a stem has a waxy layer that's even harder for the microbes to penetrate. So they're going to go on the inside. So walk out and just pick things up. Make sure it's lighter and it crushes easier.
How much harm does anhydrous do to microbes? The tightening of the soil?
Anhydrous?
Yeah, anhydrous. Yeah.
Yeah. Anhydrous kills microbes. So everywhere, I mean, you don't want to get anhydrous on yourself because it burns. Anhydrous ammonia, NH3, so one nitrogen, three hydrogens, really, really, really chemically wants another hydrogen. So it's pulling it from water. It's going to pull the water from anything it touches, including microbes. It's why we get burned when we have too much nitrogen, whether it's urea, which is just a couple ammonia molecules, or we put anhydrous down, you can burn a plant. It's because it's sucking the water out. So the things further away, just like dog spot, you probably all know what I'm talking about, where the center is dead, but the grass around it's really tall and green, those microbes are going to be fine. So the microbes will recover. You're not going to sterilize everything, but it does set them back.
Someone wants to know if the fields you showed in those pictures were new to no-till and your product had never been applied.
All of the fields were new to our product. And I want to say, because I know this guy, and by the way, I took none of these pictures and have not been to any of these fields. So these just come in from customers. They're not mine. This particular person is new to no-till. Again, because now he's saying, I won't have to use my chisel plow because I won't have so much residue in the spring, which again, too much, you all know, it can be a problem.
Here's a question from Webb. He says, what have you seen using Holganix with an extra sugar and micropack foliar over soybeans?
So all those things are just more food for the microbes. So you don't have to add extra sugar. We put a micropack in with our product, again, because it's not going to supplement a soil deficiency. There's not enough. It's just an immediate food source for the biology. But if you want to add more, it works well. The thing that we see the most that I need to learn more about, that I find really intriguing, not surprising, is when we combine with an extra carbon product. So we put a little humic and fulvic acid in our product, but people that use an extra humic or a biochar really see an even greater improvement in their soil structure and, importantly, water holding. I've had guys call me that I've never met, but they get my number from the website or whatever and say, this part of my field, I used your product and I use, such and such humic product. It's still moist. Everything else is bone dry. What's going on? And again, I go back to those biofilms I talked about. they actually break the surface tension of water so we get better wetting. Now, are you going to see that every time you use any microbial? And by the way, bacteria alone can do that. So you don't need everything else to produce biofilms. Um, there's too many variables, too much complexity. I know when I don't know, but the observations are real. So there's something to soil carbon, stable carbon, that's not the active stuff the microbes are eating, because humic acid is not a food source for microbes. It's stable carbon, as is a lot of the biochar. That plays a big role, and the microbes love it.
I'll read this next one from Olivia. She says, "Thank you for your time, Dr. Stark. As you know, regenerative agriculture aims to reduce inputs to help increase farmer profitability. In general, what is the plan for farmers to wean themselves off of bioapplications? In other words, when can soil biology become self-sustaining without bioapplications?"
Yeah, that's a great question. And again, I do this in other webinars, like I did at the No-Till National Conference there in St. Louis. Biology in the soil is constantly in flux. And to be candid, we're still learning all these details, but I know it can change week to week. It's influenced not just by the crop you're growing this year, by what you grew last year. And now we're finding that 6 to 10% of the biome in your root zone varies not just between corn, soy, wheat, and oak tree, but between corn hybrids and soy varieties. There's an intricate dance going on in the soil where the plant is actively farming microbes. It's attracting the ones it wants and repels the one it doesn't. And different hybrids and varieties have differences. I can't tell you how important those are. I can say that the soil biology is always in flux. And then winter, freezing or flooding, all disrupt the biology. Overuse of fertilizers, especially nitrogen and phosphorus, signals the plant, don't invest in the root exudates, you have excess nutrients. So that really disrupts the biology. And of course, overuse of soil fumigants or fungicides. So there's a lot going on. I can also say, though, we've got some land that's been used, with our product over 20 years. And that soil is amazing. And so we use a much lower rate, and we only do it in the spring, coming out of winter, to give balance back to the soil. So I think that as your soil gets better, you'll need a product like ours less and less. And I think that's a fantastic outcome.
All right, here's the next question. If this is added to a pre-emerge herbicide, do we need to worry about the chemical killing off the bugs?
No, actually, our product works really well with pre-emergent herbicides. And there are some herbicides, and I'm going back to biofilms again, which I think I need to start talking more about because they do so much. We have found that some herbicides, in particular glyphosate, dicamba, and 2,4-D, when tank mixed with our product, so that has to be tank mixed. And actually, I've seen this in my yard because I've got clover, and it drives me nuts, and I spray it with a Trimec, a triple herbicide. It's so waxy, it falls right off. If I put some Holganix in there with the biofilms, I control the clover. Same thing, those herbicides work better because biofilms act as extra surfactant, so we get better uptake.
Someone wants to know the cost per acre of your treatment for corn, the average in the Midwest.
Okay, so for the residue control product, it tends to be around $15 an acre. It depends on your dealer. And for the spring product, which again is similar biology, but we skew it towards the biology that really induces germination and rooting and nutrient cycling. And again, with that product, we want you to reduce fertilizer inputs because it'll work better. Everything will work better. That tends to be about $20 an acre.
Gotcha. Peter says, as compost as a result of decomposition, could I use an aerated compost tea as a replacement or reinforcement for the breakdown of residue?
Absolutely. Our product is actually based on the process of making an aerobic compost tea. That's where we get our diversity. Um, we have a proprietary source of compost in terms of what plant parts and only plant, so there's no animal or human waste in it. But unlike compost tea, and actually the inventor of our process brewed his own compost tea, and he was frustrated in batch to batch variability. It was very dilute, so he had to use a lot per acre, and it had no shelf life. He had to use it in like 8 hours. So we came up with a very different kind of process where throughout the brew, we're constantly changing conditions, food sources, etc. So we know which biology types are being amplified. So that's how we come up with a product that's consistent. We immediately cool it, it's concentrated, and it has pretty good shelf life. Out of refrigeration, 250-gallon tote will last 4 to 6 weeks. So it is like a compost tea. There's biology in there that we don't find in compost tea. I'll tell you flat out, lactobacilli. So if you like yogurt, or if you take a probiotic, lactobacilli. They're typically not found in soil, only transiently, but those organic acids are really good at unlocking nutrients that are tied up in your soil. So we are kind of a compost tea. If you like your own compost tea, great, because that's probably the kind of product I've been talking about.
If we are applying NH3 in the fall, what timing should we aim for with the bio breakdown product also in the fall to help the microbes out the most? Should we do the breakdown product after NH3 or before?
I would put the breakdown product on as soon after harvest as practical, because again, we want to extend that window where you've got temperature and hopefully some moisture. I've got some people that put it down on their corn right before harvest, and they loved it. It worked great. It's giving me even more gray hair, because what if they ran into a really, really long weather delay before they could actually go out and harvest? Remember, the microbes are weakening their stalks. And to me, that's a little bit risky. It was bold, but it worked great. So as soon after harvest as you can with the breakdown. And, I would not apply anhydrous right on top of it. I'd space it out.
What's the best soil test you'd recommend for bacteria?
That's a great question. And we are spending a lot of time this summer with our agronomy team looking at the different tests out there. And there's a lot. And they all have their strengths and weaknesses. So stay tuned. I can tell you just in general, there's nothing that jumps out that I can say no to, except you can spend a lot of money to do what's called a full metagenomic analysis. And it's going to look and try to discover all the species of microbes in your soil. And I think it's really interesting. I just don't know what to do with the data. To me, it's not about individual species with very few exceptions. It's about function. So we want to know functionally, do you have phosphorus solubilizing microbes? Do you have those that can unlock potassium? Do you have those that can act as growth stimulants? So again, and that's not a species, that's going to be broadly, because again, in God's design, there is a lot of functional redundancy. So that's the one thing I would not do. But I like the Haney test. I like microbiometer. If I didn't say your brand, I probably like it too. And we're testing all of them. So
Gord wants to know, why are seeds resistant to digestion when residue is not?
Oh, that's a great question. And the seed coat is very tough. And I do know, though, if you've got seed out in the field that's not harvested, the fungi will eventually break it down too. So I had a guy actually this morning. Now it's the cob, which also is pretty recalcitrant. He told me how surprised he was at how far it was degraded. But the seed coat is something that is designed to really help that seed obviously overwinter and and it's a good thing. That design is important for all of us.
Does applying the product result in long-term noticeable changes on the below-ground microbial community?
It does. And in terms of long term, that's the caveat that we're looking at and spending a lot of time. What we see generally within weeks is a big improvement in the fungal bacterial ratio. So, again, I'm not looking at individual species. What I really want, though, is to see how mycorrhizal fungi survive, because any kind of soil disturbance really, really sets them back. And mycorrhizae are for a number of reasons. So now, again, that's not an individual species because there's a handful of species, but now I'm narrowing in on something that's pretty important. They really get set back when any kind of soil disturbance. So stay tuned again, because again, I know we're going to see things through the season. We're going to see improvements in soil structure, but the glue I talked about that root exudates and fungal exudates and the fungal hyphae produce, bacteria are constantly eating. It's a great food source. It's like gluing together popcorn with caramel and sugar.
Oh,
They go for it.
making me hungry.
Yeah. So we need to constantly be producing that to replenish it to maintain soil structure. So I don't mind saying we're still on the learning curve, and it's not just us. I spend a lot of time looking at what everyone is doing, especially university research around the world in soil. The good news is 5-7 years ago, there wasn't a whole lot out there. Now we're, as a scientific community, starting to really pay attention to the soil. It's really exciting that we're finally learning about the most important part of our field, our soil.
So what do you think's the better option here? Applying with a pre-emerge herbicide or mix in furrow with starter fertilizer?
That's a great question. And for the last 7 years, I have looked at all the data I can and different ways customers apply the product. There is no statistical difference. The caveat is as long as you're using enough total liquid carrier with the over-the-top application to get it in the soil, which with most herbicides you are, so 15 gallons an acre, 10 is an absolute minimum. The more residue or weeds on top that you're killing with the herbicide, the better, because it's more food for the microbes. So I'd say do what's best in your operation. The other thing, again, not just our product, but with other microbial products, fertilizers are good, but again, anything that's dissolved in water scientifically is a salt. So we don't want a lot of salts with microbes. Also, microbes tend to have size. So if you've got precision equipment with like 100 mesh screens and really small orifices, and you're putting down low volume per acre, you don't want to have clogging, obviously, and I really don't want you to have clogging. You have less risk of that when you're spraying, because of course those screen sizes and openings are larger.
I think you touched on this one a little bit during your presentation, but maybe not. After corn harvest, where it will soon freeze, is there much point in applying digestion products?
I'd say no, unless you're in a place where you've got days in the winter where you're going to be in the 40s and 50s or even warmer, or the sun will warm it up. But if you're further north and you're just in winter, I wouldn't spend my money.
Gotcha. Can you tank mix with a fall applied herbicide, and what gallon water volume should breakdown be applied with?
Yeah, you can tank mix with any herbicide. And again, I like 15 gallons an acre if you can. More is better. You can even, well, you probably don't have your irrigation running, but I know people apply the product through irrigation. This is important. If you're going to use a microbial breakdown product, anybody's, don't winterize your spray equipment too early, or get your co-op or somebody to apply it for you, which more and more we're finding are willing to do.
I'm going to put my pronunciation skills to the test with this next one. Someone wrote in, mycorrhiza can break carbohydrate, polymer, and lignin?
Mycorrhizae, tend to not There are certain kinds of mycorrhizae that can actually go out and mineralize or break down organic material, but generally speaking, they don't do that. They're scavengers and they're symbionts, so they're getting fed by the plant root. So mycorrhizae are not the degraders, generally speaking, that you want. They're great for your growing crop, though, because they're really good at unlocking phosphorus. And mycorrhizae form a network through the field, and we know this from forests and such, and it's a biochemical highway. And so again, I don't want to impart intelligence to it because that's not it. It's just biochemistry. But when a plant signals a stress, an insect, a disease, they send out different biochemical signals that can then travel through the mycorrhizal network that influence the other parts of your field that then trigger a defense response in the crops before the pathogen arrives. So again, mycorrhizae are really cool scientifically. They're also really wimpy.
Here's another scenario for you. Someone says they spray their oat cover crop in the fall for preparation of no-till corn in the spring. Is there any benefit to adding this product along with fall herbicide?
Absolutely. So it's going to help the cover crop germinate and establish, as well as get to work on your residue. So it's a double win.
There you go. Well, I think we're out of time here. Those are all great questions and great answers too. And again, if we didn't get to your question, Dave's email address is right there at the bottom of your screen, dstark@holganix.com. And I bet you'll get a lot of emails from this session, Dave.
I hope so.
Thanks so much. That was a really great presentation, just jam-packed with information. The hour just flew by. And a reminder, if you missed anything or you want to go back and watch the presentation again or share it with someone, we'll email out the recording of this entire webinar within about 24 hours or so. We'll also have it posted on the no-tillfarmer.com website and No-Till Farmer YouTube page. So on behalf of Dave Stark and Holganix and No-Till Farmer, thanks so much for joining us for today's webinar, and hope to see you for future webinars, and have a great day.